So you should have reviewed the first two, or the first learning objective rather, to learn the general anatomy of skeletal muscle. And then the second and third objective, we're going to talk about later on towards the end. So just to kind of run through the things that you should be highlighting, the structure of muscle. Definitely make sure you're comfortable with the structure of muscle, the three layers of muscle, the terminology that we use that's specific to muscle. So the plasma membrane here is the sarcolemma, the cytoplasm is the sarcoplasm. Also think about the overall structural organization of muscle. This is really important as we think about the function of muscle and then the sarcomere specifically, kind of where that is in the general organization of the muscle itself. So while we think about the layers, right, the epimysium, perimysium, endomysium, also think about the compartments. So the muscle fiber itself, which is within the endomysium, the fascicle, which is a bundle of muscle fibers, and then the entire muscle being the bundle of fascicles together, right? So that structural hierarchy is important. Also know the structural organelles, so the subcellular components, things like the sarcoplasmic reticulum and how it functions with storing calcium. That's going to come back when we talk about the regulation of muscle contraction and how that calcium gets released from the sarcoplasmic reticulum, and then it can interact with actin, and then it can engage and allow for the shortening of the sarcomere. Okay, similarly with the T-tubules, this is also likewise important because it helps the action potential go throughout the muscle or spread throughout the muscle very quickly. So definitely think about the importance of those structural components. Now, when we get to the sarcomere itself, so think about a myofibril, and then think about the repeating units on that myofibril, and then that is where we have the sarcomere. So the sarcomere is formed by these two really important filamentous proteins, actin and myosin. And the importance of these two proteins is that they bring about the shortening of the sarcomere, which brings about the shortening of the myofibril, the shortening of the muscle fiber, and the shortening of the entire muscle. So these are essentially the functional units of the muscle itself. When we get to the histology and the banding and staining patterns, definitely know what structures are formed within each of these bands and zones. So for an example, the I band is only thin filaments or actin filaments, right? So that's what's going to be a little bit lighter staining. It's also going to be around the Z line. So if you look at the histology at the bottom here, we can see that the lightest staining region of the sarcomere is where we have the thin filaments only, which sort of makes sense, right? They're smaller proteins, they're less dense, they're going to stain less densely on histology, as opposed to, say, the A band, right, which is the central part of the sarcomere, which has an overlap. So the A band has an overlap of thin and thick filaments. That's another unique feature of the A band. And so it's going to be darker in its staining density because it actually has two proteins, forming this region. And then the M line being the very center where the thick filaments are adjoined, or the myosin filaments are adjoined, hence the M here for M line. And then the Z line or Z disc is another important part of the sarcomere. Specifically, the Z line is going to be the borders of the sarcomere. So when we think about one sarcomere, it's from one Z line to another Z line. And so this space within here is a single sarcomere, and that is a repeating unit that is happening along the length of the myofibril. All right. So all of that is likewise important, what exactly these different bands and zones are representing. And then we move on to look at the structure of actin and myosin independently. So what actin looks like independently, we talked about how it has the actin monomers itself, which is the blue structures here that line up like pearls on a string almost. And then we have the two regulatory proteins that are associated with the actin filament, which would be tropomyosin and troponin. And really important to distinguish that actin is the contractile unit. Actin is the only unit that is moving necessarily. Tropomyosin and troponin are only regulating the movement of actin. So they don't have any contractile property in and of themselves. They are basically supporting the contractile unit, which is the actin strand or the actin filament. So really important to think about the role of tropomyosin as basically covering up the binding sites on the actin monomer, right? Whereas troponin is going to allow for the binding of calcium. It's going to allow for the binding to the actin unit at the bottom here. And then the third site will allow for the binding of the tropomyosin monomer, the tropomyosin thread-like protein. So that when calcium binds to troponin, the entire troponin complex can shift its state, and that will drag tropomyosin off of those binding sites and effectively expose those binding sites, which are the black dots here. And that would allow myosin to engage with actin and drag actin towards the M line. All right, so definitely think about how these regulatory proteins can turn muscle contraction on by exposing those binding sites and turn muscle contraction off by covering up those binding sites in response to the absence or presence of calcium on that troponin specifically. Okay, then there's myosin. So this is the other protein. This is where these actin binding sites will engage onto actin itself. And so we want to think about the myosin as the energized filament. Actin is sort of playing a passive role here, and myosin is the one that has the ATPase binding site, which is where it can be energized by binding ATP. And so that is going to help in the dragging of the actin filament. So the filament that is doing the dragging is the one that binds the ATP, that has the energy, and that would be the myosin or the thick filament. Okay. And then this drawing is really just a summary of kind of all of that, all of what we just said, but more specifically to think about how this is functionally, right? So how do we actually shorten this sarcomere? And so it's basically the myosin heads reaching up, grabbing on to the actin. So the actin is in red here. The myosin is in blue here. So this myosin is going to grab onto the actin and then drag this towards the M line. Oops, towards the center here. And so that will shorten, as the Z lines get closer to each other, that will shorten the entire sarcomere and then eventually shorten the entire muscle, right? That is how we contract muscles. Okay. The other important thing I'm going to jump right to is the cross-bridge cycling. So this is another important aspect that hopefully you reviewed, basically thinking about the cycle here of how that shortening takes place, how the two heads of, or the two states of the myosin heads are allowing for binding to actin, dragging actin towards the M line, an event that we refer to as the power stroke. So that dragging of actin towards the M line, what we call the cross-bridge cycling, is basically bringing that actin towards the center of the sarcomere. And then when the new myosin binds onto this, or the new ATP binds onto this myosin head, then it releases the myosin head from the actin, and then we can start the cyclical process. Again, the myosin head goes into its cocked state, its high energy form, and then it's basically waiting for calcium to come in, expose the binding sites, and then once we hydrolyze that new ATP, we can reattach to the actin filament and then begin this process again and again. And this is happening cyclically in order to bring the actin filaments closer and closer and closer. So it doesn't just drag actin all the way to the M line. It's a cyclical process of dragging, releasing, dragging, releasing, dragging, releasing, depending on exactly how short that sarcomere needs to be, which is really determined by how much force that muscle needs to generate.